Hello students and welcome to today's lesson. Please go ahead and pause your screen to answer the following bell work questions. Co-activators are another type of way that we can communicate with cells. So the target cells end up having a response to a lipid-soluble cell signal can vary enormously depending on the nature of the cell. So basically it's saying that the target cell's response to a signal that is lipid-soluble is going to change the type of response or the signal transduction, and many different signals are able to happen with co-activators, and even the same cell type may have different responses to a lipid-soluble cell signal. It depends on which co-activators are present to change the type of signal transduction pathway that occurs within the target cell. For example, estrogen has different effects in uterine tissue than mammary tissue. So these are both types of tissue, but they have different effects on the different tissues because of the types of co-activators that are present. It is not the presence or absence of the receptor, but the presence or absence of a co-activator. So now we're going to get into talking a little bit about co-activators. So here are different types of cell surface receptors or membrane receptors. So we have chemically gated ion channels, enzymatic receptors, and G-coupled protein receptors. Chemically gated ion channels is a channel that is linked to a receptor. So it's a gate that doesn't open unless the receptor ligand complex is activated. So after it becomes activated, it allows a specific ion to pass in response to that complex being formed. Enzymatic receptors is where the receptor is an enzyme that's going to speed up a chemical reaction that is activated by the ligand. So the ligand enzyme process is what is going to activate the enzyme. Almost all of these enzyme type receptors are protein kinases. We also have G-coupled protein receptors, which is a G protein embedded within the cell's plasma membrane, is bound to a GTP that is within the intracellular space. So it's a protein bound to a GTP, which is bound to the cell membrane, and this assists in transmitting the signal from the receptor to enzyme. Enzyme is the effector that will take place after the signal transduction pathway is initiated. Okay, so here is our pictures to help you figure this out. The first picture has our ions and an ion-specific channel. Our ligand is the green substrate, I guess the green shape, right? After the ligand is bound to the receptor, it forms the ligand receptor complex, and therefore it allows the ions to enter the cell. That is where our signal transduction pathway can take place, so the cell is going to elicit some response from these ions entering the cell. Okay, and here is our ligand receptor activating the enzyme. So we have the ligand binding to our receptor, which activates the enzyme and allows for the cell to respond. So whatever this enzyme is allows for a chemical reaction to happen inside of the cell. Here is our GDP, our G-coupled protein reaction. That's what GCP stands for, G-coupled protein receptor. The ligand binds to this G-coupled protein receptor. then this G protein is able to elicit a different response within the cell. These both on the bottom are part of the G-coupled protein reaction. So after our ligand binds to our G-coupled protein receptor, it allows for the enabling of the GDP protein, and that GDP protein can then react with the ion channel, allowing solutes to pass that are channel specific, and then GDP becomes GTP from this binding, and the other part of the GTP can then go and react with an enzyme to allow for chemical reaction to take place. Chemically gated ion channels. So we have this very commonly happening with neural transmission. So you have a presynaptic cell and a postsynaptic cell, and this chemical is the chemical gated channels can be voltage gated channels because they aren't going to open until a specific voltage is reached within the cell. So we have synaptic vesicles containing neurotransmitters that are going to perform exocytosis into the synaptic cleft. The synaptic cleft then will be touching a second postsynaptic cell, and these ions, or the neurotransmitters rather, that are released into the synaptic cleft can bind to the postsynaptic cell's receptors that are specific to that type of neurotransmitter, and they are gated, ligand gated ion channels. So after the neurotransmitter binds to that postsynaptic membrane, it allows for the passageway of those ions that are specific to that channel, and then allows for entrance of those ions into the synaptic space, or into the postsynaptic cell. Okay, receptor kinases. Protein kinases phosphorylate proteins to alter protein function. So these kinase enzymes are going to have a phosphorylation and then alter the protein's function. So we have receptor tyrosine kinases, which are RTKs, and they influence the cell cycle, cell migration, cell metabolism, and cell proliferation. And the alteration to the function of these protein receptor kinases can lead to cancer, because we have basically uncontrolled cell proliferation, cellular growth, which is what cancer is. We have uncontrolled cell growth that it's no longer performing its function of stopping reproducing cells. Receptor tyrosine kinases are membrane receptors, and plants possess receptors with an overall similar structure and function. So here is the activation of the receptor tyrosine kinase proteins animation. So you have inactive monomers, and then after they bind to their receptor, they dimerize. So dimerization occurs when these two receptors basically form a dimer or come together. After dimerization, we can have ATP become ADP and phosphorylate these protein kinase receptors. And that is going to therefore activate certain proteins through these protein kinase receptor complexes. So the monomers bound to the protein kinases plus dimerization plus ATP is going to have the ability to activate these relay proteins within the intracellular space. Here is another picture of it. Basically everything I explained. One, the ligand binds to the receptor tyrosine kinase proteins. and following the binding of the ligands to the RTK protein receptors, they dimerize, and autophosphorylation occurs through the addition of ATP, which then becomes ADP. And this allows for the phosphopiranese receptor complexes to bind and phosphorylate other proteins as well, which changes the function of these proteins. All right, insulin receptors. So this is how the RTK can work to lower blood sugar. Insulin is a hormone that helps to maintain a constant level of blood glucose. Glucose is bound to other glucose molecules and converted into glycogen, and this lowers the blood glucose by taking the sugar from the blood and converting it into something that the cell can use for storage. So we have 1, insulin binds to the extracellular domain of the subunit of the insulin receptor. So we have a receptor and ligand complex. The insulin receptor complex is therefore bound and activated. The subunit of the insulin receptor phosphorylates the other, allowing the insulin response proteins to be activated. So insulin is the receptor there's an insulin receptor that allows for the entry of glucose. Phosphorylation of insulin response to proteins activate the glycogen synthesis. So because of this protein complex, this receptor ligand protein complex, we are able now to take the excessive glucose that is within the blood and convert it into glycogen so we can store it within the cells. Because glucose alone cannot be stored in the cells, we have to convert it into glycogen. Okay, here's a summary of our receptor tyrosine kinase complexes. A single transmembrane domain anchors RTKs into the membrane. Extracellular ligand binding domain is where the ligand is able to bind to the extracellular component of the receptor complex. There's an intracellular kinase domain, which is a catalytic site of receptor that acts as a protein kinase to speed up the chemical reaction that is to occur within the intracellular environment. Process. When a ligand binds, dimerization occurs and autophosphorylation occurs, changing ATP into ADP. Adenine triphosphate changes into adenine diphosphate, and a dimerization occurs where the two different receptor complexes come together to form one larger receptor complex. The cellular response follows, and it depends on the cellular response of the proteins within the intracellular space. Okay, everyone, here's your exit ticket for the day. Go ahead and pause your screen so you can answer this question and complete the week's participation points and bell work. Have a great rest of your day, and be sure to stay on the lookout for announcements regarding the new cell communications or body mechanisms group assignment.